Okay. All right, everyone. Dr. Phil here. It's June 4th, 2021. I'm going to redo chapter 7. I can't find it, so I'm going to record this into WebEx, and then I will pop it into the course via YouTube, I think, just so it won't take up space. So I want to go over chapter 7 on nutrition, and then I'm going to do chapter 8. It'll be on exam 1, and I'll kind of go through and just kind of let you know what is probably important or what they're going to probably be asking you in the future. So let me see if I can get this to work here. I'm using a different PowerPoint. This is for assisted learning, so it's going to be a little different. So bear with me if it doesn't work perfect. All right, so microbial nutrition. So just realize that microbes have to have nutrition the same as we do, except they don't have a digestive system. So they're going to be bringing nutrients from the external environment in, or if they're in our body through the interstitial fluid, whatever they're in, and they're going to be bringing it in. So sometimes they can't really digest exactly what's out there. So they may have to send out enzymes or enzymes or lysomes to go in and break things down and then reabsorb them in a different form. And we talked in lab, and I think I mentioned it in lecture, that they are going to have chemotaxis too. So they're going to be drawn towards a chemical gradient of something they want. And if they can move, we can generally assume that they have a flagella, some kind of way of moving. So in lab last night, we did endospore staining and capsule staining, but just realized that endospores or capsules, they don't help them move at all. They just happen to be virulence factors or ways survival of the fittest, so the species can survive. So anyway, we'll get started. So nutrition is a process by which chemical substances or nutrients are acquired from the environment and used in cellular activity. So this can be for eukaryotic or prokaryotic. All right. So this, I'm assuming, is review, but essential nutrients must be provided to an organism, which means that they can't produce them, or they have to have them. And we'll talk about micronutrients and macronutrients, what they need in big quantities or small quantities. We'll talk about certain growth factors that these need if we're going to be growing bacteria in a lab, or if we're incubating it. We have to be able to provide all the essential nutrients they need, or they won't be able to survive. If they're missing any one nutrient at some point in time, they will perish. Same as us. If we don't have an essential nutrient, we won't be able to produce specific proteins, and we won't be able to produce specific enzymes. And then at some point in time, we'll die. There'll be a disease process going on. So macronutrients, macro always means big, required in large amounts, play an important role in cell structure and metabolism. So these would be your proteins and carbohydrates, same as us. We basically, the majority of our food is probably proteins, carbohydrates. Lipids would work too, but bacteria don't really do a whole lot with lipids. All right. And even if they did, they would just consume carbohydrates and make their own lipids. We as humans, we need lipids to store essential A, D, E, and K essential vitamins, and we need lipids for cell membrane production. We need lipids for microglia, for cell wall structure. We need for like bubble wrap around the orbit of the eye, the kidneys. So they're kind of packing in the body, and we won't get into brown fat or white fat or whatever. So micronutrients and trace elements required in small amounts involved in enzyme function and maintenance of protein structure. So these are going to be helping the ribosomes and everything make these specific proteins and hold them together. And these would be things like magnesium, zinc, nickel, these trace elements. And we'll see in the Krebs cycle, we're going to need some of these trace elements also. Okay, it is working. Okay. All right. So there's from your book, just kind of showing you kind of what we're going to be talking about today: pH, nutrients, gases. So whether something is aerobic, obligate, facultative, or whatever, and just realize that sunlight really produces all the energy on Earth. So we can talk about photosynthesis and how it produces plants, and then animals eat plants, and then we eat the animals, and we can do the whole photosynthesis reaction, and then we can do the Krebs cycle reaction. We can run that reaction back and forth. It's the same equation, we just run it back and forth. All right. We'll talk a little bit today about temperature, like what organisms, extremophiles or some organisms like, or they have a niche market, very, very high temperature, very, very low temperature, like listeria likes low temperatures, or it can grow in low temperatures, so it can grow in your refrigerator. Theoretically, it can grow on ice cream, things like that. We'll talk about popping up. Sorry, it's my birthday today, so you may be getting some Facebook pop-ups. Anyway, we'll talk about salt. Some organisms can live in high salt concentrations. We'll talk about pH, and we should know that neutral pH is 7.0. So in the 6.8 and 7.5 or 6 range for most organisms. Heliobacter, well, we know can live in the stomach. It can survive in a lower pH, but it also can it can induce bicarbonate in its surroundings, so it can actually raise the pH a little bit, but we'll talk about that. Okay, so organic nutrients contain carbon and hydrogen atoms and are usually the products of living things. So methane, that's just a simple carbon. This is crazy. All right. So methane is carbon with 4 hydrogens around it. Carbohydrates. That's a carbohydrate. C6H12O6 is always going to be that 1-to-1 ratio. Lipids, proteins, and nucleic acids. These are all organic nutrients. Combination of carbon and hydrogen. Inorganic nutrients are atom or molecules that contain a combination of atoms other than carbon and hydrogen. So anything that's other than carbon and hydrogen. And when we talk next chapter about the Krebs cycle, we'll see like what is the final electron acceptor of certain organisms. It could be oxygen, or it cannot be oxygen. So if something is, aerobic or anaerobic, it could change what the final electron acceptor is for that Krebs cycle. So metals and their salts. And we talked yesterday in lab about Graham's iodine and crystal violet and how they actually produce a salt inside of the peptidoglycan layer, making it bigger, making it harder for that larger molecule to leave. They went in as single positive and negative charges, and they combined. All right. It's chemical analysis of cell contents. So just like humans, prokaryotic, eukaryotic, 7% water, proteins, 96% of the cell is composed of 6 elements: carbon, hydrogen, oxygen. So those should be pretty fundamentally, you should be knowledgeable about fundamentally. Phosphorus, sulfur, and nitrogen. Some others. Right. So here's from your book, just really showing you sources of these and why are they significant. All right. So these, just kind of read through that and just kind of jog your memory of all that. Sodium, we should know that it regulates osmotic pressure, and water always follows sodium. And then we'll talk later about, theoretically, if I took a red blood cell and I put it in plain water, I put it in salt water, or put it in something that was isotonic, same 0.9% sodium chloride, how would it affect or affect this. And then we'll talk later. We'll talk about carbon when we talk about endospores and how they can have like a calcium carbonate shell around them. And we talked in lab yesterday about how they found endospores from a long time ago. So theoretically, like from a mummy in the pyramids, they could bring these endospores back to life from 10,000 years ago. Right. So sources of essential nutrients. So these are all, but they have to have a carbon source, right? So all organisms have to have a carbon source, and it can be a carbohydrate or it can be a sugar, but there are some bacteria, when we do the Enterobacteriaceae lab at the end of the semester, we're going to find there's a bacterium, I'm not going to tell you what it is right now, that can literally live, it can find any source of carbon. It doesn't have to be a sugar or fat or what we're used to, the C6H12O6 or fats. They have carbon, hydrogen, and oxygen, but the ratio might be slightly different. All right. But a heterotroph must obtain carbon in an organic form made by other living organisms, such as proteins, carbohydrates, lipids, and nucleic acid. So they have to obtain carbon from something. And like I just said, it could be a carbohydrate or lipids, it can be protein. It's just like with eukaryotic cells, it's a little harder for them to pull the carbon off of protein. They'd rather use carbohydrates and possibly lipids. All right, autotroph. All right, so that's self or whatever, an organism that uses carbon dioxide and inorganic gas as its carbon source. So that actually can take the carbon right out of the CO2. And these things, the cool thing about them is they're not nutritionally dependent on other living things. They'll just pull it out of the atmosphere. So growth factors are essential organic nutrients. All right, and I think I talked about media before, and how there's certain specific media, and it has to have certain growth factors. And if any of those growth factors are not in the media, whether it is a agar plate or a broth media, the organism will not live. I can give it the perfect conditions as far as temperature and oxygen or lack of oxygen whenever it needs. But if I don't have a specific growth factor it needs, it can't reproduce. So organic compounds that cannot be synthesized by organism because they lack the genetic and metabolic mechanisms to synthesize them. Same as us, we have certain essential amino acids that we have to consume. We cannot produce them. All right. So growth factors must be provided as a nutrient, essential amino acids and vitamins. And I just kind of, so I just pulled these out. These are just broth media. This is a nutrient broth. So it has generally what most organisms need. And then I have, I'll put this up here. I don't know if you can see that. So this is brain heart infused broth. All right. The cool thing about this is we don't know exactly what's in the brain and heart that the organism needs, but we just know that it has growth factors, and it has essential nutrients for some fastidious bacteria. And I kind of learned from one of the best that if you have a bacteria that's a little sluggish, doesn't really want to grow, she told me, yeah, put a little brain heart infused media, and sure enough, the organism thrived in that. All right, so nutrient types. Many determinants of nutrient types are, what's its carbon source? Is it a heterotroph? Does it need to consume other organisms or other carbon sources from outside the environment? Is it going to send out an is going to send out some sort of digestive enzyme and break down something in the external environment and then bring it back in a different form? Or is it an autotroph? Can it actually take the carbon, like we said, right out of CO2? So a chemo or chemo, whatever you want, however you want to remember this, gain energy from chemical compounds, right? Phototroph, troph means to eat, really. Trophosite or troph. get energy through photosynthesis. They're going to actually pull it out of the sun. So on your exam, I put a bunch of these in. It's going to auto-generate whichever question you get. So it'll be something about what is a photoautotrope? What is a chemoautotrope? And the name just tells you exactly what it is, okay? It's a saprobe, right? Metabolizing the organic matter of dead organisms. So we're talking about like a dead tree or something, that would be saprophobic bacteria would live on that. Parasite, though, they really kind of want something living. They want to really hijack. They're really lazy. They just want to hijack something living and just steal its resources. Instead of a parasite in your digestive system, it's so lazy, it wants to be in your digestive system. You're going to keep it warm, you're going to keep it protected from the environment, and you're going to provide all those nutrients for it. You just have to lay there and reproduce, right? Photoheterotroph, all right? So these sunlight organic matter, this is really what the purple and green photosynthetic bacteria are, and they're really, like I said, they're archaea. So they're not going to be important for us specifically in healthcare, is that they're not going to find us as a host very hospitable, right? So photoautotrophs, these are oxygenic photosynthesis or anoxygenic photosynthesis. I mean, does it need oxygen? Can it live without oxygen? All right. Chemoautotrophs, it tells you exactly what it is. All right, so it survives totally on inorganic substances, and chemoautotrophs, remember, they can just pull their carbon source from anything. Methogens produce methane gas under anaerobic conditions. Oh Lord, what have I done now? Oh, I guess that's how I swipe the MacBook. All right, so heterotrophs and their energy sources. So remember, heterotrophs, they have to get their energy from somewhere. So majority are chemoheterotrophs using aerobic respiration. The two that we really care about are the saprophobes. So I said before, these are free-living microorganisms that feed on organic citrus from dead organisms. So this would be, you know, they could live off maybe a carcass or maybe a tree or something that wasn't living, or, you know, I guess theoretically leather or something like that, right? So these can provide an opportunistic pathogenic problem if they're living off your dead necrotic tissue, right? You have dead tissue in a wound and they're living off it, right? That would be saprophobic, theoretically, right? So they're a facultative parasite. But true parasites really derive the nutrients from the host. These are pathogens, and some of these are obligate parasites, all right? And that literally means that they are so lazy and so dependent, they actually have to go from host to host. So we'll be talking later on about some of these sexual transmitted disease like chlamydia and things like that. They literally have to go from host to host. They can't even live on a surface for more than a small amount of time. So there's a question in your exam. You'll get some form of passive active transport or some facilitated transport. So just have an idea. This should be review from anatomy, physiology, or biology, or some other course you've taken, but we're going to mention it again here because we're talking about eukaryotes and prokaryotes. This is microbiology. So we're talking about things that are super tiny. They're not going to be great big things like mammals, but you know, if we're talking about the roundworms, or cestodes, or nematodes, or some of these you know, larger vectors or these bacteria, they still are going to have the same metabolic processes, maybe slightly different, but the general idea is the same. So passive transport. You know, you're talking about, took an A&P, we're talking about passive action or active, like muscle. Is it passively doing this or actively doing it? Does not require energy if it's passive. Substances exist in a gradient and move from areas of high concentration towards areas of low concentration. So that's basic diffusion. Right? So we're talking about, usually diffusion is strictly should be water and a diffusing membrane. But always remember, it's always easier to go from the top of the stairs down. So it's always easier to go from higher to lower. It's not easy, and it's against the chemical gradient to go from lower to higher. So that always requires energy, some form. Right. So osmosis is diffusion of water. Right. Facilitated diffusion requires a carrier. Okay. So that just means it might be whatever we're diffusing might be too large to get through normally, so it needs something to carry it, facilitate it, and a facilitator to carry it through. All right, but it's still passive. In active transport, the name tells you, requires energy and carrier proteins. It's gradient independent. It's not dependent on the gradient, it's independent of it. So active transport. All right, and a good example of this would be something in your like in your kidneys. Okay, so the proximal convoluted tubule, that's when things leave the glomerulus, things that the body wants to reclaim are going to be very easily diffused through the chemical gradient. But if we really want to move things against their chemical gradient, we'll see we have to use active transport, and that takes a lot of energy. So that's why the kidneys have a lot of mitochondria to produce ATP, to produce energy. That's active transport. So we're going to be moving things against the concentration gradient. We're going to go from low to high. Group translocations, transport molecule chemically altered. So if we're bringing something into the organism, and we already know that's going to have to be turned into a compound or a molecule, we just save the energy in a step. We just, as it's coming in, we attach it and then bring it in changed. So rather than bringing it all the way in and then finding the other molecule and causing it to combine, we know what's going to happen, so we just group translocate the whole thing. And then bulk transport, endocytosis, are we going to be pulling in nutrients from the outside of the environment, or pinocytosis with water, or exocytosis, if we're going to be getting things, things leaving the cell. So whether it's a protein or an enzyme, or waste, like if we phagocytose something of the immune system or a macrophage, phagocytose it, we digest it, we're going to reclaim whatever we can use. The rest of it we're going to exocytose outside to the interstitial fluid, and then hopefully our lymphatic system will take it back into our blood. The kidneys can process some of that, and the liver will process some of that too. Right. So diffusions, the net movement of molecules down their concentration gradient, so passive transport. So this is sort of kind of showing you like a sugar cube in hot water, or if you're making like my acupuncturist gave me some kind of Chinese tea. So I put that in hot water and I stir it, and then I walk away, and then 10 minutes later it's diffused. So whatever's in that tea, or if it's sugar or anything, over time things will bounce and they'll move. They're always in motion, and they will keep moving until everything is equally separated. So everything's diffused equally in the space. So whether it is in a gas or in a liquid, it's going to bounce around back and forth until everything is completely evenly distributed. Oh my, this is so secretary. Shit. Oh, this is going to be awful. Okay, let's get through this. So hopefully this will play for you guys. It doesn't play on the MacBook, but it should play specifically if you go use Google and you go with a regular non Apple product, it should work. Right. So they're also showing you this diffusion here. So in anatomy physiology or your human bio, they should have taken a like a kidney diffusing membrane, kidney dialysis tubing, put different concentrations of sugar in there, so 20%, 40%, or 60%. Glucose or sucrose. Sucrose is like table sugar, so that's much cheaper. Or you could use salt too. They put it in the diffusion membrane, you put it in a beaker of water. All right, so theoretically, the higher the concentration of salt or sugar, over time, it's going to pull more of that fluid in to equalize the osmotic pressure, and then you're going to weigh those after a certain amount of time. So whether it's 10 minutes, 20 minutes, 30 minutes, an hour, doesn't really matter. But if you weigh the 20% diffusing membrane, the 40% the diffusing membrane, or the 60% with the diffusing membrane, the higher the concentration, the more they should weigh, which means they pulled in more water. And remember that what's going to happen is water is always going to go towards the solution with the most solutes in it. It's going to really, it just tries to equally distribute all of that solute somehow. Right. So here you guys can review that. So if something is isotonic, it's the same, 0.9%, more or less, inside the cell and outside the cell. So there should be no exchange, all right? But if it's a hypotonic solution, that fusion of water is into the cell, right? This swells the protoplasm and pushes it tightly against the wall. Wall usually prevents cell from bursting, but if we have enough water going in, it'll occupy the space and it'll burst. And that's what we really want to do with a gram-positive bacterium or any bacterium. If we can break down that peptidoglycan layer, which helps protect it from the external environment and the osmotic pressure, we can but through diffusion, fluid will go into the bacterium and then it'll literally burst it, okay? Hypertonic solution, water diffused out of the cell, it shrinks the cell membrane away from the cell, plasmolysis or crenation. So let's say we put a cell, red blood cell, in seawater, right? So seawater, hopefully we know that the sodium concentration is higher than inside the cell. So I said before that water is always going to go towards the area of higher solute, all right? So what happens is the water from inside the cell is going to leave the cell and try to dilute all around it to make that solute, that sodium concentration equal. All right, it's a losing battle, but the red blood cell doesn't know that. All right, facilitated diffusion. This is passive transport. Right. So this is it's going to bring these large molecules in passively. Doesn't require any energy. All right. But if we were talking about the sodium potassium pumps, they're moving against the concentration gradient. They require a lot of energy. Right. And if you were in pathophysiology or maybe a slightly more advanced course, you'll find out they have hydropic change is when we can't get enough blood to perfuse the tissue. So an ischemia or some kind of problem, we can't get enough blood there to bring oxygen to run the Krebs cycle, so we run out of energy. So what happens is the sodium potassium pumps don't have enough energy to run, and the osmolarity changes. Right? So these cells they start swelling up, they causing a lot of problems. And if we can fix the problem before the cell dies or ruptures, it's fine. If we can't, then we have hydropic change and then we have cell death. So here's carrier-mediated active transport, just bringing these molecules in. right? And here's the group translocation I was talking about before. So we already know that we want this protein. We know that this is going to combine to make the protein at some point. We just know that, or the cell knows that. So it's thinking, well, why don't we just save a step? We'll put this component here, and as we bring them in, we'll just attach them and then bring it in. Rather than bring them both in and have to recombine it over here, we're just going to bring it all the way in. It's group translocation. I'm hoping the graphics work on this. All right. And then endocytosis, eating or drinking of cells. Endocytosis is really a substance through a vesicle or a phagosome. Phagosome is something that it's a vesicle that goes and engulfs something and then it consumes it. It uses whatever it wants and then it gets rid of the rest. Right? So ingest substances or cells, and penocytosis ingest water molecules. So it really goes, and just realize the pseudopods go out, they wrap around it, they connect it, and they bring it in so that at no point in time is the organism, inside of the organism, exposed to the outside environment directly. It always does it through this cell membrane of the cell. So once again, there will be a question or two on active passive transport on the exam. I don't know, I think there's maybe 18 questions on that. So when you go to click in, it's going to randomly generate something for you. So environmental factors that influence microbes, a niche or niche market is a totality of adaptation organisms made to their habitat. So they're really organisms that find an environment where only they can survive. So they may have a genetic mutation, or they may have a virulence factor, they may have some kind of adaptive ability that they may not even have known about. They get to an environment and they actually can adapt to it. And they just they replicate, they have a quorum sensing issue, they start replicating, and then they realize, well, nothing else in the area can live in that environment, so they have a niche market. There's no competition, right? The issue kind of becomes after the niche bacterium or whatever it is lives there for a while. As they ferment or as they change things, they start changing the environment. And if the new change in the environment allows another organism to come in and grow, then we can have another organism on top of that one or living commensurate with that. And then we have the potential of having a biofilm generating. All right. So environmental factors affect the function of metabolic enzymes. So same with us. I mean, our environment will affect our metabolic capacity. And as our metabolic capacity changes, we may need to produce enzymes to build up, break down, digest certain things. So remember, enzymes are factors that help things connect much faster, but they don't really participate in the process themselves. But they help things link or set up much faster. All right. So functions include temperature, oxygen requirements, pH, osmotic pressure, or barometric pressure. So these can either deter it or help it. Right? Right. Explain that later. Yeah, metabolic enzymes. Okay. Yeah, and enzymes, I want to bring that up too. So, we can talk about for a second. We can talk about denaturing proteins, right? Everyone knows you denature proteins, things will die, will change, right? But please realize if we denature something, change the pH or something like that, we generally affect the enzymes first. And as we affect the enzymes, metabolism can really be distorted, slow down, or disrupted completely. So that's really how denaturing proteins works. So really, it starts with the enzymes first. Okay, we're not going to really denature huge proteins as quickly as we're going to denature the enzymes. So the enzymes would be the first thing to be affected. All right. So cardinal temperatures, or these are kind of law or groups or categories. So minimum temperature is the lowest temperature that permits the microbes growth and metabolism. Right? So it would be the lowest temperature. For us as humans, it depends on what that is, but at some point in time, we wouldn't be able to survive, right? Maximum temperature is the highest temperature that permits a microbe's growth and metabolism. So if we're talking about bacterium, they really can't produce a whole lot of heat energy, and they can't go to Walmart and buy a coat or whatever. So they really kind of want to live like between that 20 to 42 range. Anything below that, the enzymatic processes start to slow down to a screeching halt. All right. Or anything above that, the temperature is too high. It really starts denaturing their enzymes. So that also causes their metabolic processes to come to a screeching halt. Right. Optimum temperature provides the fastest rate of growth and metabolism. So for us here in lab, the incubator is set, I think, at 36 or 37 degrees. All right. So things really thrive in there. I could leave something on the counter. Overnight, it'll grow at room temperature. But if I put it in the incubator where it's nice and warm, that stuff will grow overnight. It'll cover a complete plate. So actually, I brought this. So today is June 4th, right? Yeah. So that is a regular plate, nutrient agar plate. This I wrote on here, staff for the practical. I made this June 3rd. I'll just contaminate that. So that's the difference. Nothing overnight growth. All right. So psychrophiles, their optimum temperature is below 15 degrees Celsius. They're capable of growing at 0 degrees Celsius. So these only one I can think of that would be close. Like I said, it was listeria that can grow lower. It can a little bit lower than its optimal growth, but it could. Mesophiles, so these are what usually is the human pathogens, or anything that's going to affect us, or anything that we're talking about here that would be potentially a human pathogen or human disease causing, even if it was a opportunistic organism, like to be between 20 and 40. All right. Thermophiles, all right, optimum temperature is greater than 45 degrees Celsius. So these are the microbes that live in hot tubs or hot springs, or they live some live at the bottom of the ocean, where these volcanic vents come and the water is literally boiling and the pressure is super high. So there are like barophiles and thermophiles. Crazy. And if you bring that thing to the surface, you get rid of that pressure, it'll just open, die. Oxygen. As oxygen is utilized, it is transformed into several toxic products. All right. So this is extremely important that we realize right here and now. If an organism has no way of dealing with oxygen, it's toxic, even for us. Oxygen is toxic. We need it, but it's toxic. So we have to have a way of dealing with it. All right. The organism has no way of dealing with it, it will eventually die. So even like Clostridium perfringens that causes gas gangrene. All right, it's anaerobic. It doesn't like oxygen. It has no way of dealing with it. So if we debride a wound and we start hitting it with oxygen, the organism will die. All right, or like that's how hydrogen peroxide works. Even if an organism has a way of dealing with oxygen, they can't deal with that concentration of hydrogen peroxide. Or if we leave hydrogen peroxide in contact with the organism long enough, it will kill it. Right? And that's actually theoretically, you could kill even endospores with hydrogen peroxide with high enough concentration, long enough exposure. Theoretically, you could do that. But I was talking to students last night in the lab, and a couple of them actually were in dental hygiene. And I asked them if any of them gargled to use hydrogen peroxide in their, what are those things called? Water picks, right? And they said yes. And I said, well, why? Well, that's because they told us to do that. Well, here, the deal is that the hydrogen peroxide will kill most of the strep, mutans, or viridans on your teeth, right? And you'll actually feel it foaming, so you know it's working, right? Or if you have an infection in your finger and you pour hydrogen peroxide, you'll just see it bubble, catalase production. You'll see it bubbling. And the side effect of using hydrogen peroxide, by the way, is that it might just whiten your teeth a little more too. But anyway, so singlet oxygen super ion or superoxide ion peroxide, or hydroxyl radicals. So these are all ways that these things oxygen is transformed into several toxic products. Right? We have a way of dealing with that. Most cells have developed enzymes that neutralize these chemicals. So superoxide dismutase enzyme, ends in ASE, or catalase, like Staph aureus is catalase positive. It's a definitive way of knowing it's Staph aureus as opposed to Staph epididymis or something else. If a microbe is not capable of dealing with toxic oxygen, it is forced to live in oxygen-free environments. So that's where all the anaerobic bacterium live, or they could be facultative. You know, they could live in small amounts of microfacultative. They could live in small amounts of oxygen, but they don't really like it. And we'll see when we do our Enterobacteriaceae lab, if we stab into our tubes, we'll see that all the growth is more towards the bottom. Even if it's on a slant tube, we'll see, well, there's nothing grew on the top of the slant where most things would grow. I don't bring a slant with me here. But for some organisms, they're living below the stab line because there's no oxygen there. They like it. Or Clostridium difficile, you probably all heard of that, C. diff. All right. You know, we'll talk about wiping out the microbiome later and how it overtakes it. But C. diff lives in the large intestine, all right? And it lives there for a reason, because the large intestine has the least amount of oxygen, right? You know, as you go from the stomach, the duodenum, ileum, and so on, there's still oxygen in there. But as you go towards the distal end of the GI tract, there's less and less oxygen. So Clostridium difficile is anaerobic. All right, so it loves that environment. All right, so aerobe utilizes oxygen and it can detoxify it to some degree. All right, obligate aerobe. All right, this is, so something's going to be aerobic or facultative anaerobe. They can utilize oxygen and they can detoxify it. Other things, they have to have oxygen. They can't grow without oxygen. They're obligated, obligate aerobe. Facultative anaerobe utilizes oxygen, but it can also grow in its absence. So anaerobic, they can grow in the dearth or the non-presence of oxygen. All right. So, but facultative means they have ways of dealing with it. All right. Micro aerophilic requires only a small amount of oxygen. So these generally live, if we were doing experiment with a slant or a tube, they would grow right at the very, very top. And generally they'd be just under the surface, right? They only require a small amount of oxygen. They really don't want to be out in the atmosphere where there's a lot of oxygen. All right, anaerobe does not utilize oxygen and it can't, it'll die in oxygen concentration. All right, and I'll show you guys later again, but I also did a strep. They prefer a little less oxygen, so I did it in one of these CO2 chambers, and if you look in there, I actually lit a candle, all right, to pull all the oxygen out of the environment. And we'll see how my my strep plate came out. All right. So obligate anaerobe, but they're obligated to not be around oxygen. They lack the enzymes to detoxify oxygen, so they cannot survive in an oxygen environment. And autotolerant anaerobes, they don't utilize oxygen, but they can survive and grow in its presence. So it doesn't, they don't use it, but they're autotolerant of it. So here's culturing by oxygen requirements. And I said before, these are slant tubes, right? So if it's like a semi-solid gel. If we stabbed, do I have one? So this is like an oxidation needle. So if I sterilize this and I pick up part of my culture, and this was a gel tube, and I drove it down in there and pulled it back out. Anything that grew down here, if this was a gel, would be anaerobic. It would be away from the oxygen. All right. And then anything above here would have just died because there's too much oxygen for it. Right. And right here, all right, there's a chamber. They pull out the oxygen out. All right. So these microbes here, it's probably strep because it looks like it's in a blood agar plate. They don't like oxygen, or they like it diminished, so they're going to grow there. Right. Also, carbon dioxide requirement. All microbes require some carbon dioxide in their metabolism. Capnophile grows best in higher CO2 tensions than normal were present in the atmosphere. Right. So something that would grow in here would be a capnophile. It grows best at high CO2 tensions normally, but strep will grow in oxygen environment. It goes in the back of your throat, right? Okay. Majority of microbes grow at a pH between 6 and 8, so these are neutral. And we know the pH range goes from 0 to 14, okay? So the middle would be 7, that's neutral. And if a pH is 7, it means it has the exact amount of hydrogens, molecules, or yeah, the H plus is exactly the same as the OH minus, all right? They're completely equal, all right? If there's more H than OH, that's going to be slightly acidic. If there's more OH than H, it's going to be slightly basic. You know, acidophiles, they grow at extreme acid pH, so that would be like Heliobacter. Alkanophiles grow at extreme alkaline pH. Most microbes exist under hypotonic and isotonic conditions. All right, so remember, hypotonic means that the amount of solute is lower than normal. Isotonic means that the number of solutes are the same as inside the cell. Iso means the same. Right? Halophiles require high concentrations of salt. Right? So this would be something that if it requires it, can only live in higher concentration. You know, we could say like Staph aureus or Staph epidermidis, they can live in like up to a 7% salt environment. So if we had a I can't think of mannitol salt plate, and we swabbed it, the only thing that would theoretically grow on there would be, because it has a 7% salt concentration and some mannitol alcohol. That's selective and differential, but either one, I digress. ADHD. But those plates will inhibit the growth of anything else other than Staph aureus or Staph epidermidis, because nothing else can grow on that plate because of the high cell concentration. Right. Osmotolerant do not require high concentrations of solute, but they can tolerate it when it occurs. So osmotolerant, that means they don't require it, but they can tolerate it. All right, baryophiles can live under extreme pressure and will rupture if exposed to normal atmospheric pressure. And I was talking about before about some of these thermophiles. They're like high temperatures at the bottom of the ocean or wherever there's some kind of volcanic vents in the seafloor. the temperature is super high, right around that, and the pressure is super high. So the barophiles, they like be under a lot of pressure. If you bring them to the surface or takes away some of that pressure, they just pop. They need that pressure to hold them together. So just sort of know symbiotic or non-symbiotic. So symbiotic organisms live in close nutritional relationships required by one or both members. So if you're living in symbiotic, if you have a symbiotic relationship, you you both sort of benefit, except for parasites. So if mutualism, so obligatory, dependent, both members benefit. So if you have a mutualistic symbiotic relationship, that's good. You know, you both live together, you both sort of need each other, and both benefit. It's kind of how the first first eukaryotic cells developed, right? They had a mutual symbiotic mutualistic relationship. One consumed the other, but the one that consumed the one was protecting it against the environment, and the other one said, well, you're protecting me, you're doing all the work, I'll just make all the energy. Right. Commensalism, the commensal benefits, others, members not harmed. So you're living commensally, one organism benefits, the other one's not harmed. So some organisms, they they might feed off digestive waste of one, so they're benefiting off the digestive waste of the other one. The other one's not harmed by that at all. But parasite is dependent on something else for its energy, and it's draining or taking away from the organism. Right. So non-symbiotic organisms are free living relationships not required for survival. So they don't require any other organisms for their survival. Right. So synergism, members cooperate and share benefits, and this is seen in a lot of biofilms. Right. And antagonism, some members are inhibited or destroyed by others. And this is kind of what you want in your, it's not really a good example, but this is what you really want in your microbiome. You want organisms there, their being there inhibits or destroys or keeps other organisms at bay. So, everyone says, well, gut bacteria is disgusting. Well, you need gut bacteria to help digest your food. You need it for vitamin K production. You need it for a lot of things. And it helps keep way worse things away. Way worse, that's not even a word. All right, so mutualism is obligatory, dependent, both members benefit. Mutually, it's good. Commensalism, like I said, commensal member benefits, the other member is neither harmed nor benefited. And parasitism, the host is always harmed. So ecologically, what does this mean? So the non-symbiotic organisms are free-living, relationships are not required for survival. Right? So it's not a symbiotic relationship. Synergism, members cooperate to produce a result that none of them could do alone. So this is with a lot of quorum sensing in some of these biofilms, and that's really like 1 and 1 is 4. So everyone has a certain part. None of them would do it alone. So if we were talking about so if we're talking about even if you're setting up a village somewhere in the pioneers or whatever, they would have a synergistic relationship where members cooperated to produce something. So they had to, like, they wouldn't be able to survive on their own. So everyone had to pull their weight and do something very specific. All right. So like socialism wouldn't work in that environment. All right. So everyone had to kind of do their own kind of thing. So antagonism, action of one organism affects the success or survival of the other. All right. So, all it would take would be one member to destroy something or to consume all of. You're talking about microbes. If one consumed all the oxygen or one consumed all the carbon source, the others would die. Right. The synergism, you know, one is everyone's sharing the load of whatever's going on. They all contribute something for survival of the fittest, to advance the species or just advance the group. So human body is a rich habitat for symbiotic bacteria, fungus, and a few protozoa. This is normal microbiome flora. So you have some bacteria, literally have some fungus, and you might even have some protozoa growing in your digestive tract. Not a big deal. All right. All right, there is, I think this should run, it's in Blackboard. But if you just kind of want it, a few minutes, learn about the microbiome, it's fascinating. All right. So biofilms result when organisms attach to their substrate by some form of extracellular matrix. So it could be a slime layer, it could technically be fimbriae. So they literally attach. So they're the primary residence. So they're going to attach there. They're going to be living there, fermenting. You know, if it's on your teeth, they're fermenting the sugars in your teeth, producing acid. They're changing the pH a little bit. They're excreting some waste. Another organism comes along and says, "Oh, well, we may not love this lower pH of this slightly acidic environment, but we can survive around it. And you know, we like some of that waste that you're expelling. We could use that." So they grow on top of that. Another one goes on top of that, and before long, you have a bunch of different organisms growing in a biofilm or a layered environment. Now, the cool thing is, as these other organisms grow on top of it, they're protecting everything underneath it, right? So the dominant structure of most natural environments on Earth. So we've been talking about, you know, if you look at rocks, yeah, especially around water, like more in Canada, I've seen like stuff growing in rocks. It literally breaks down the rocks and stones into soil. So these are things that are growing, could be, like I said, on dead logs or something. They're growing in the environment. Communicate and cooperate in the formation and functions of biofilms. So quorum sensing, it really kind of means that once everything is all good and they realize that the environment's great, everything's going to be working, they kind of send out a signal. Okay, guys, everything looks good. Start going through binary fission and let's reproduce. So free swimming cells settle on the surface and remain there. So these cells are here. They're kind of going to so just, oh, this is not going to work. It worked before. All right, so they're going to come down here. They're going to start they start synthesizing a sticky matrix that holds them tight with the substrate. So they're going to lay down a layer that's going to cause them to stick there. Right? When the biofilm grows to a certain density or quorum, like if you were talking about parliamentary procedure, you're voting on something, you have a quorum, you have a certain amount. All right. Cells release inducer molecules that can coordinate a response, and they just really signal everything to start growing. All right. And then as these grow, other organisms come along and they start growing on top of it. So microbial growth occurs at two levels. Growth at a cellular level increases the size, all right, increase in population. So if we're talking about going through binary fission, you know, the cell grows and then it sort of divides in two, so they're two identical, and then those two are small, and they're going to grow. So we have the growth as in we go from 1 to 2, but those two have to start growing before they can divide into 2, right? Division of bacterial cells occurs mainly through binary fission, and that's in the transverse plane. All right. All right. So if we were talking about in lab, we were talking about strep, staph, and like one's going to be linear in a chain. Okay, the other one's going to be in clusters. So it depends on how they go through binary fission. If they're in one plane, two planes, all four X, Y, and Z plane. So the parent cell enlarges, duplicates its chromosomes, and forms a central transverse septum, all right, and divides the cell into two daughter cells. Similar here. So we have a grown cell. Oh, I'm sorry, this is initial, just went through binary fission like down here. It grows in size, then it makes a septum, divides chromosomes, kind of like an embroidery hoop, replicates, opens up, now you have two identical, divides, and then we have two cells. And these also have to, remember, these have to grow into that size before they can replicate too. So time required for a complete fusion, fission cycle called the generation or doubling time. So most bacteria that we're going to be dealing with, it's like 20, 30 minutes. Every 20, 30 minutes, they go through binary fission. So they reproduce logarithmically. So each new fission cycle increases the population by a factor of 2. It's exponential growth. Generation times vary from minutes to days. And if we're talking about like microbacterium or some Yersinia that cause bubonic plague, well, no, I'm sorry, like leprosy or TB, microbiosis, these go through binary fission every couple of days or longer. So their generation time is very, very long. And please realize that antimicrobials only work usually during the dividing and growth phase. So course of treatment for them is a very, very long time. All right. So equation for calculating population size over time. All right. I could give you guys a question say I have for bacterium and their generation time is 35 minutes. In 3 hours, how many would you have? All right. There's really no point in learning that, but just realize that things replicate logarithmically. That's the important key here. All right. Here's a viable plate count. So we could just say, if we could literally count this, we could say, you know, we started with this. 120 minutes later, we got this. 180 minutes, we got this. 240 minutes later, we got this. 300 minutes later. So you see, as we go through here, logarithmically, they're increasing incredibly fast. Okay. So if we took like this, let's say we inoculated this. All right, we inoculated it with 1,000 E. coli. All right. And then came back a couple hours later, I was this color, I'd say, oh, well, something's growing in there. Right. And if I came back and I came up with this, see how turbid that is? I would say there's definitely something growing here. The only problem we have with this is I knew something grew, but I don't know if it's alive, theoretically, unless I played it, because I can't take its pulse or respiration, right? So I can't see if it's breathing or take its pulse. So I don't really know. All right, so this is going to be important for growth, and this is going to be really important for staging of diseases, and it's going to be important for any kind of microbial, whether it's detergent or if it's antimicrobial. In laboratory studies, populations typically display a predictable pattern over time. This is the growth curve. So, you know, we'll get into this here in a minute. Lag phases, we just inoculated this tube. We have to give it a while to get acclimated to the new environment, and it's got to figure out, well, is there quorum sensing? Is there enough information? Do I know it's going to be a good environment? And then once we get the, okay, we're all adjusted, everything's good, there's enough food, the oxygen concentration is good, start reproducing. All right. There's very little growth here. All right. Then we get to the exponential growth. So everything's great. And we start going through binary fission logarithmically. We go, wow, 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 great, awesome. Keep reproducing, keep reproducing. There's plenty of food. There's plenty of space. There's plenty of oxygen, whatever. Then we get to a certain phase here where we're saying, well, wait a minute. we reach our limit. All right, so as some die off, we have to replace it. So it's very stationary. So the death rate and the growth rate are the same. And then at some point in time, they realize, guys, all the dead bacteria are piling up. We're living in our own waste. We're out of food and oxygen. So they start dying off. Theoretically, that's how it works. So exponential phase here, once again, a period of maximum growth will continue as long as cells have adequate nutrients and favorable environments. And we can see that right here. Okay. And then the stationary phase. So the key with this is the rate of cell growth equals the rate of cell death. Like I said, they're depleted in nutrients and oxygen. They're living in their own excrement. Not good. And then as limiting factors intensify, cells die exponentially. So at some point in time, all the oxygen is going to be used up, and they're living in all of their waste products. And turbidity, like I said before, this is the easiest way to tell. Degrees of colonic turbidity reflects the relative population size, but like I said, this, we can say theoretically is 0. Right. All right. I know something's growing, it's good. When it gets to this, we're like, okay, definitely something growing. And at some point in time, this, if I left this in the incubator for whatever, a month or whatever, it could possibly potentially just be dead. We use up all of the waste. And we can just tell the difference by just shining a light through it. But once again, I said, we can't take its temperature. We can't take its pulse or respiration. We don't know if they're all alive in there. I can't say that everything in there is alive, but I can say something grew. I can at least say that. Okay, I give up. What in the hell? Okay, I am going to assume this is the end of 7. All right, let me process this, and I will pop this.